The mouse. This tiny creature has had a huge impact on science research. Mice make excellent models for human disease because parts of their DNA are similar to human DNA. They suffer from many of the same ailments as humans, like cancer, diabetes, and even anxiety. Research on mice has helped scientists understand the causes of many different diseases, and the use of brain imaging with mice has been a big boost to studies of brain function in humans. But mice brains are tiny. And guess what? Imaging a little brain for research isn't easy. Now, scientists may have found a better, easier way to help further research.
Inside Science.
To Adam Bauer, this little creature is an amazing research tool.
So the beauty of imaging mouse models, or even rat models, or monkeys, or people, is that all of us, all mammals, have hemoglobin in their brains.
Hemoglobin is the molecule in blood that carries oxygen. It makes it easier to take an image of the brain because it works as a good contrast agent during a functional MRI scan, helping to light up certain parts of the brain.
That's something that we can all image, from humans all the way down to mice. The fact that mice exhibit these functional networks that resemble very closely to what we see in people is really powerful.
Just like doctors use functional MRI or fMRI to look at human brain activity, Bauer uses a similar technique, but for tiny mouse brain activity. The technology shows which area of the brain is in use during a particular mental process while a mouse is awake.
What we develop is kind of an fMRI surrogate for use in mice. So up until recently, fMRI was having a pretty difficult time creating really nice images in the mouse brain using the standard techniques that they use in people. It's come a long way, and they're able to generate some really beautiful images now, looking at changes in the blood activity in the brain. The mice will be brought into our imaging systems, and we'll image their activity. So there'll be very specific tasks that we'll have these mice perform. So certain regions of the brain are responsible for the execution of specific tasks.
Bauer also looks at the brains of mice that have been given a stroke. Researchers can learn how the mouse brain works to restore functional areas and remap itself to compensate for the damage from a stroke, and translate those results for restoring more function in humans.
And so by imaging mice, you can learn a lot about human disease and how to affect it and treat it and make it better in people.
This is Inside Science.
Inside Science. If you enjoyed this edition, follow us on the web and social media. Powered by the American Institute of Physics and a coalition of underwriters.
Good morning, one and all. Myself, Gigi KN. It's my privilege and pleasure on behalf of the management, principal, and staff of CL Baitmeta College of Pharmacy to welcome all the registered participants and Honorable Speaker of the Day, Dr. C. Mayuran, to our international webinar entitled Ethical Consideration in Animal Experimentation and Research. This webinar is organized by Department of Pharmacology jointly with the Department of Pharmacy Practice of CL Baitmeta College of Pharmacy, Chennai. We thank our management and the beloved Principal Madam for their constant encouragement and support, and also providing us an opportunity to conduct this webinar. Before starting the session, I would like to give a brief introduction about our speaker. Dr. C. Mayuran is currently working as a program director for BPharm Honors Senior Lecturer at School of Pharmacy, International Medical University, Kuala Lumpur, Malaysia. It gives me immense pleasure and proud to inform you all that Dr. C. Mayuran is an alumnus of our college. He completed his B Pharmacy and M Pharmacy from C.L. Mehta College of Pharmacy in the year 2003 and 2005 respectively. He pursued his doctorate degree in pharmacy from Vinayaga Mission University in the year 2010. He is having a total of 15 years of academic and research experience. He is a recipient of 6 research grants from various government nodal agencies of India and Malaysia, also serving as an international collaborative researcher with many foreign universities. So far, he had published around 50 research articles in various national and international journals, published one book, and guided 33 research projects. He is a member of reviewing panel in various international journals, also the member of editorial board for Asia Pacific Journal of Health Sciences, Indian Journal of Pharmaceutical and Biological Research. In the year 2017, he was honored with the Best Researcher Award in Diabetes Asia Conference that was held at Kuala Lumpur, Malaysia. In the same year, he also received the Best Oral Presenter Award in ISCOMS that held at Netherlands. With this brief introduction, once again I welcome you, sir, for the session. Now the session is yours. Kindly proceed.
Thanks, Dr. Gigi. Hello, everyone. Very good morning. I hope you all are sound and safe in this pandemic situation. This is Dr. Mayuran from International Medical University, Kuala Lumpur, Malaysia. It gives me immense pleasure to be part of this webinar series organized by C.L. Mehta College of Pharmacy, Chennai, India, which is also my alma mater, and I'm proud of it. Thanks to Professor Great Ratnam, Principal CLBMCP, Professor Muralidharan, Head Department of Pharmacology, CL Mehta College of Pharmacy, Professor Krishna, Head Department of Pharmacy Practice, CLBMCP, for extending an invitation to deliver a talk entitled Ethical Considerations in Animal Experimentation and Research today. I would also like to take this opportunity to thank Associate Professor Dr. Mohammad Zulkifli, Dean, School of Pharmacy, International Medical University, Kuala Lumpur, for granting approval and providing support to deliver this session. Before we begin the talk, a brief introduction about myself and the programs at IMU. IMU has 36 programs as of now, and I am the program director for the Bachelor of Pharmacy Honors Program at IMU. The BPharm Honors Program is a 4-year program which can be done 4 years at IMU, or also has options to take 2 years at IMU and also fly overseas to New Zealand and take the rest of the 2 years at University of Otago. Or another option would be to go to University of Tasmania, Australia for the last 2 years, because we do have the credit transfer options. Without further ado, we shall now begin the session. All right. Okay, may I know if you are able to see my slide?
Yes, sir.
Yeah, we are able to see.
Okay, can we start?
Yes, sir. You can purchase.
All right, thank you. All right, so regarding the title for today, what are we going to expect, or what you can expect for this session would be an introduction, a very brief introduction to animal experimentation, followed by what are the practices or the species that are used, and how this ethics came into use, and the chronology of how the institutional animal ethics committee, that particular the regulations came into implementation. Then what is the meaning of 3 R's? All right, and what are the roles and responsibilities of a researcher being adopting the ethical principles, and then revolving around the human practices, and finally the alternatives to animal experimentation. So today's session will cover, if not in depth, very briefly going to the time factor. So we will move forward. Okay, what is animal experimentation? Because since this topic is focusing on research, it is very necessary to know what is animal experimentation, what are the things associated with it. To my knowledge, as far as literature is concerned, the protocol, the ethical principles adopted by the Germans, the German animal welfare is supposed to be the most stringent body which governs these practices. We do have all these principles and regulations in many countries, but as per the literature, it says the Germans are very, very strict. Okay, so according to the German Welfare Act, the experimentation can be defined as animal experimentation as interventions and manipulations in animals if this is associated with suffering, pain, and injury to animals. Okay, so taking these factors into consideration, this applies to all procedures or protocols which will subject the animal to stress. Okay, so that is the reason why we need to have something in place to govern, okay, or to regulate. Regulation is very, very important. Unless and until it is regulated, it cannot be streamlined. Okay, it cannot be refined further. Okay, so that is the reason why in reality, it means that every procedure or each procedure carried out on the animals or animal models for scientific purpose, it must be recorded. It must be recorded as an animal experiment, and it has to be approved by the respective governing body. Okay, so you need to look at the global perspective first, then we narrow it down to the Indian perspective. If you look into the global perspective, every individual country, they have their own governing body at the national level, then that will cascade it down to the institutional level. Okay, so this at the national level, several policies were there, and eventually the cascading to the institutional level came down later. Okay, moving forward. So as mentioned earlier about the regulation and the processes. So as a component of the approval process, the protocol should explain the reason why are we using animals, okay, and what are the measures that we are going to take, okay, in regard with their living, in regard with their care, etc. So all these things are part of the approval process itself. Okay, so the approval will be necessary for different types of research. So this list here gives you some idea about the different types of research in which we use animals. Okay, one is basic research, which is inevitable or very much necessary, okay, to move forward. So applied research for prevention. Okay, this is somewhat different or an advanced type of research relying on the basic research. Then to another area, detection and treatment of diseases, quality testing. Other than this, we also have promoting of animal well-being, improving the housing facility, conservation of species, training, etc., professional CPD programs. So in all these things, you might have to use animals, right? So be it any sort of area where you're going to use animals, it has to be approved. That is the basic concept. Okay, moving forward, use of animals in research. Okay, so if you see majority of the animals in research, they are around 31 or 31.1% focusing on basic research. Basic research, as I told you, the basic elements which helps you to gain some data in order to move forward. Okay, so around 11.9% is actually contributed by the translational and applied research. So this data is also relying on the data that was available in the literature, referring 5 years down the lane in Germany. Around 28.2%, okay, they are not actually exposed to experimental treatments, but they have been put down for the sake of extracting cells because we are moving towards replacement. So usage of cells or cell culture, we call it as primary cell lines, which will eventually be modified to immortalize cells, right? So for this purpose, they are all put down. So around 23.7% have been used for safety checks. Okay, so this is only from one of the literature, which is not the global percentage, it is from one single country. Okay, so talking about animal species, so animal species used in research are mainly small animals. The small animals like the mice, rats, guinea pigs, rabbits, all these things are example of small animals. So from these small animals, if you see the major contribution or I mean, for most of the experimental part, we will be using mice or rats. They are classified as commonly used animals. Okay, and they are also sacrificed for extraction of cells. Okay, so why are they becoming very common, or what is the reason behind it? If you look into the mice model or the mouse genome, the mouse genome has been completely studied, okay, and several manipulations have been done on it. So which will be we will be able to make use of that data to mimic the human condition. So it is not necessary. I mean, if you are able to come up with some data using the mouse model, then you will be able to extrapolate it, okay, or translate it, because translation is very important when you come to research. Okay, you cannot just okay, I've done a study. It's just for my own benefit. I just throw the data. No, the data is very valuable. It can be used anywhere and it can just move forward. So you need to adopt the right model there. Okay, practicing all the ethical principles. Okay, once this, moving on from this phase, now we have moved towards using fish. Okay, the use of fish has tremendously increased as well. Okay, when this usage of fish has increased, the data says that the usage of the rats and mice have declined a bit because now the genome of the zebrafish have been extensively decoded. Okay, and they are able to provide great insights into the life process in vertebrates. So the animal size is becoming smaller as well, and the quality of the data is improving. So you might have to play very safely which one you would want to adopt. Okay? So in practice, as mentioned earlier, we are talking about basic research. Okay, so the basic research, the ultimate aim of the basic research is to gain some very important knowledge. Okay, or we call it as some preliminary data. Okay, or studies. This basic research has no immediate application. Okay, you cannot immediately translate it, but this basic research would be very important to provide scientific evidence, okay, to move forward in the same area. For example, my area of interest is diabetes, and when I would want to go for a funded project, I need to have some data because I need to prove in my funding application that this research really works because I'm going to ask for animals, so I need to justify. Okay, so because of the similarity between humans and animals in terms of their metabolic processes and function of organs, knowledge gained in animal experiments can provide a better understanding of life processes and their disturbances in both humans as well as in animals. Okay, so talking about the basic research, although the transferability of the results from basic to its application cannot be planned, and its direct short-term benefit. Okay, if you are looking for something very immediate, then it cannot be predicted. Okay, scientific and medical breakthroughs are not conceivable through this basic research without the knowledge gain from basic research. Right? You need this data, but you cannot say that okay, the research stops here. I've been able to gain adequate amount of evidence to ensure that this will work in humans, or it's just going to work in human models. No, it's not possible. All right. So what I want to emphasize here is basic research is still important. We cannot ignore it. Okay. So now from the basic research, we are moving to something called as advanced. Okay, advanced exploration. Okay. So this medical research or applied research, otherwise we call it as, is inextricably linked or highly knotted, okay, or then very closely knitted together with basic research. A very good example that I would want to give here is the development of the treatment for diabetes. Okay, so way back in 1920s, insulin was identified as the hormone that regulated blood sugar. So experiments have been conducted on rabbits, dogs, pigs, cows. These experimentation help to understand the effect of insulin on blood sugar levels, and this contributed to the development of new therapies. Okay, which eventually worked on human models. Okay, so rabbits, dogs, and other mammals have been largely replaced by rats and mice in physiological research these days. Okay, because the extent of data that could be collected and the validity of the data is very important, as I mentioned. So when you can rely on the data that you have gained with this usage of smaller models, you will have to think again whether it is necessary. Okay. Another another advantage here is the rapid reproduction of these species. Okay, permitted specific breeding for individual clinical presentations. This includes, for example, diabetes mouse. Okay, with elevated blood sugar levels, etc. So you need not go into the chemical induction of diabetes. You can just procure those rodents which are already diabetic. So the data that you collect out of that research will be very much useful, okay, and valid as well. So this is how the basic techniques are linked to the advanced research. Okay. So other than this basic and medical research, we also have other research in which animals are used. So that includes all the list that I have given here: diagnostics, extensively, yes; transplantation medicine, because organ transplantation, the organ rejection studies, okay, replacement with animal tissue, stem cells is a growing area where animals are used. Genome research, such as I mentioned about mice, about the fly, the fruit fly, and the zebrafish. They are all the current advancements in science. So how we were able to get into that, we will have to still rely on some basic research. Okay, so extensively used in neuroscience, veterinary research, etc. Okay. So these are the other areas where we still the experimentation on animals are continuing. Okay, so moving forward about the evolution of ethics. So the reason why I spoke about all these animal models and to what extent they are used, what is the percentage areas and all, is to understand that there is a need for it to be regulated. If it is not going to be regulated, it is just going to be exploited. And be it anything, whether it's vertebrates or invertebrates, or whether it's small or big, pain is pain. The feeling is going to be the same, right? So you need to take that component into consideration while you are engaging on research. Ethical practice is never to be ignored. Okay, so you see, if you look into the data that I have presented here, we are talking about research now, we are talking about ethics now, but you look into the century, it has dated back to 2nd century itself. Those days itself, the Greek and the Romans, they have referred to rules, regulations. Okay, so the earliest reference to animal welfare and ethics is dated in the 19th century. I mean, you might be knowing we have CPC, SCA in India, and we have also SPCA and all. So SPCA, if you look into the literature, it is dating back to 1822. Okay, so it is not somewhere where all these things started now. It has been existing, but only thing, it became very stringent and it was cascaded down to the institutional level a bit recently. Okay, so several principles or ideologies have been followed. Okay, for example, virtue ethics, okay, deontology, utilitarianism, okay, abolitionism, all these things. Huh? So what does this principle talk about? Okay, for example, your contractarianism speaks about acceptable if most people accept the experimental objectives without offense. So this was the principle or ideology or the theory behind that particular uh, uh, ethical practice. Okay, so if you go into the deontology, so this says any form of beneficence towards humans has to be weighed with the non-maleficence towards animals. So you need to weigh without any wrongdoing to the animals. What is the benefit? Okay. So about the utilitarianism, the theory behind that is acceptable if adequate human benefit is accepted. Okay. So it is like that. So all these principles or theories existed. Okay, so from that time forward, then came into the institutional level. Okay, so we will look into the chronology. So before going into the chronology, so this is something to know about institutional animal ethics committee. Okay, so the institutional reviewing. Okay, so it is a committee which exists at the institutional level, which has to be reporting or which will be reporting to a governing body at the national level. Okay, so the goals of the reviewing committee would be setting guidelines for ascertaining whether animals are necessary for a study in the first place. You need to think whether you need to have animals or not. Overseeing humane experimental conduct, okay, whether it is adopting all the humane principles, okay. Specific tailoring of methodology is very important. You want to do something on, for example, diabetes, then you need to choose the right model. So species specificity, because if the species specificity is not there, right, your data is not reliable. So you might have to repeat the study again and again in order to get a reliable data. So you might want to use more animals, which is not agreeable at the first place. So you need to ensure. So establishing minimum discomfort to experimental animals. So these are the different goals that or the strength that will include the institutional ethical committee to review the protocols. Okay, so the four running nations include Australia, then Sweden, Canada, the US, we have India, and the UK. So we have got rules, regulations. Okay, but when those rules and regulations have been cascaded to the institutional level to ensure that there is adequate amount of recording, monitoring, reviewing in place. Okay, so if cascading down to the Indian perspective, if you look into it, we had the prevention, or we still have the Prevention of Cruelty Act, which is available from 1960. But in 1998, then the CPC, SCA, which is the governing body at the national level, did some further modifications of the regulations, incorporating the breeding aspects and experimental part further, which eventually cascaded down to institutional level. Okay, so this is how we will have to look at this one. Okay, so talking about the reviewing process, etc., it is very necessary that the ethics committee or we as a researcher, while developing a protocol or reviewing a protocol, we need to look into principles. Okay, so what are those things? So although animal experimentation cannot be completely avoided in research, there is a general consensus that it must be restricted to necessary minimum. Okay, so the 3Rs principle devised in the year 1959 by William Russell and Rex Burr can be taken as a guideline or adopted as a guideline for animal experiments. Okay, so which has been universally recognized. Okay, so the 3Rs, what do they stand for? They stand for refinement, reduction, and replacement. Okay, so the aim of the principle is to avoid animal experiments where possible. So the consistent and responsible implementation of this principle accommodates the ethical concerns against the use of animals, and it also will help to improve the quality of the test results. So that is why we have these ethical considerations. Under the ethical considerations, we have this 3Rs principle. Okay. So talking about one of the R, that is refinement. Okay. So the ultimate objective, if you look into it, minimizing the adverse effects of research. Minimizing the adverse effects of research. Okay. So this will actually bring into the amount of suffering or the capacity of the animal to undergo any form of suffering. Okay, so this capacity actually depends upon the neuronal development. Okay, so if you look into it, the strongest protection should therefore be given to animals presumed to have greatest capacity for experience. Okay, due to their advanced stage of development. For example, we can take primates as a very good example for this one, because when the animal model, you are going for a larger animal model, of course the suffering will be huge, so you need to think. Okay, so it doesn't mean that since you are taking the larger model, you'll have to look for it, and when it comes to a smaller model, you can just leave it like that, not like that. So refinement not only attempts to reduce the stress in animals, but promotes positive mental and physical states. Okay, the refining could be associated with many things. For example, housing, how you perform the protocol, handling, etc., etc. Okay, so refinement has to be taken into consideration. So, carefully selecting the animal model, alleviating pain using analgesia, anesthesia, improving the technology used in the measurement procedures, developing non-invasive research methods, okay, has been possible to make with all these sort of approach. It has been possible to make great progress in the refinement of animal experiments. Example, rodents are provided with nesting materials that help support their regulation, okay, the thermoregulation, or paper rolls that are given, which are placed inside their cages, okay, because it is also a kind of bedding material. These improvements are also in interest of research as they improve the reproducibility of experimental research, because once the stress factor is refined, the reproducibility will be improved, because your data will be altered if the animal is under stress, or if you are not following the right protocol, be it anything. The protocol could be housing, could be experimental part, could be data collection part, anything. So the refinement has to be looked upon panoramically. You need to have a bird's eye view on it. Okay, where are the options for us to refine? Then talking about the reduction component. Okay, so talking about reduction, okay, you cannot just think that, okay, we are supposed to reduce, I'm supposed to use less number of animals, otherwise the ethical committee is going to come back to me. So you need to look at the statistical significance. There is something called as power analysis. And if you are a researcher or if you are a pharmacologist, you need to ensure that you have the right animals in your group, because you need to have significance. Okay, you need to have right amount of number which will give you significantly valid data. Okay, so the balance of statistically significant numbers and minimize the number of animals should be established. Okay, so statistically and methodologically improvements help to reduce the number of animals that we can use in our experimental model. Okay, so it is very necessary as a researcher. The researcher will have to ensure that this balance is existing in the protocol, and the researcher has ensured that he has looked into all possible ways of reducing, and this is the number that he or she wants, and this shouldn't affect the statistical significance. So this has to be justified to the committee that you are presenting your research. Okay, so next is about replacing. Okay. So wherever possible, replacement has to be adopted. Okay, replacement meaning that, for example, in vitro techniques. In vitro techniques are very much useful, as I mentioned earlier, about animals being used for cell cultures, right? So those things will have to be taken into consideration. You will be able to collect some valid amount of data, because with my experience, while I was applying for my research, when you apply to ministry-funded research, your protocol will not be, or your proposal will not be accepted unless and until you have some valid data with you. So that valid data could be adopted from cell cultures or smaller animal models, or using a smaller number just to give them some confidence, or computer modeling, etc. So wherever possible, it has to be done. Otherwise, so if the research question permits simple organisms such as bacteria, invertebrates, cell, tissue cultures, computer models, all these things are alternatives will have to be considered. Okay, otherwise there'll be a question whether have you considered or not. So if you have not considered, then there might be a very big question why. So it's better that you approach in a better way. Okay, so these are the principles. So these principles are also adopted by the CPCA guidelines. If you look into the CPCA guidelines, the principle 1 talks about advancement. I mean, you are permitted, or you can claim that I'm doing this particular research with the help of animals to advance or to embark on a new discovery. Okay, to know some to improve on the knowledge or the information that is available on the physiological context. Okay, all the data that is going to be produced or collected is going to save and prolong human life. Okay, or it is going to bring down suffering, be it animals or be it human beings. So it should be finally be used. Okay, so that is the first principle focusing about. Again, the second principle, the scale, okay, the degree of sentience, so which is about the talking about the phylogenetic scale, the feeling experienced or perceived. Okay? So that should be considered. So animal lowest on the phylogenetic scale and still be able to give scientifically a significant result should be considered as the first priority. Okay, so and then comes the alternative. I mean, the other models. If it is not feasible, alternatives not involving animal testing should be given consideration first. And if you were to use the other models, then you will have to justify. So principle 3 is avoidance or minimization of pain and suffering, which is considered as the priority by all means. So proper usage of sedation or accommodating sedating principles, techniques, analgesia, anesthesia wherever possible should be adopted. Okay. The principle 4 is about the welfare of the animals. Okay, so when you're focusing on the welfare of the animals, it involves everything, right from housing, experimental protocol while you are doing the experiments, the aftercare, rehabilitation, whether you're going to reuse it or whether you'll have to euthanize it, means sacrifice the animal using human practice by following human practice. That is very, very important. So this information, which I'm not going to read out everything, which is self-explanatory. So these are the conditions whereby euthanasia can actually be considered, because our primary aim is to minimize suffering and pain. Okay, so you will have to look into all these principles. So principle number 5 is talking about the housing, feeding, and care of all animals used for biomedical purpose must be directed by a veterinarian or other scientists in the relevant discipline, who is trained and experienced in the proper care, handling, and use of the species being maintained or studied. Okay. So in all circumstances, the veterinary care is very, very important, because you need to ensure that the living conditions of the animals are appropriate all the time and their health is taken care and they are comfortable. Okay. So this goes down to the level of even changing the bedding. Sometimes, me being a pharmacologist, I know it's not that easy to actually work inside the animal house, but inevitably, since we are into research, we will have to changing of bedding. All right, so you need to ensure the bedding is clean, etc., etc. The animal, the water is free from pathogens, and it is tested. Okay, the temperature. Okay, the windows, the lighting, everything will have to be taken into consideration while you are following the principles, and you need to adopt it, because you as a researcher, you have no choice. Okay, or people working with animal models have no choice. Right. Now, talking about the uh, the humane endpoint. So before we talk about the humane endpoints, it is very necessary that we need to recognize uh, the pain and the distress that the animals undergo. So the ultimate purpose of the application of human endpoints to toxicological studies is to be able to accurately predict severe pain, severe distress, suffering or impending death before the animal experiences these effects. Okay, so it is very necessary that all the individuals in this particular setup are aware of these guidelines or practices. Okay, so obviously the guiding principles says that earliest possible endpoints will have to be taken into consideration. Okay, so the earliest possible endpoints, you will be able to come to this endpoints with the help of other informations. Okay, you cannot just find out the endpoint immediately, but you will have to rely on literatures. You will have to rely on previous studies. Okay, the studies could be pertaining to anything. For example, you are using a chemical, and if you have a similar structure, another chemical entity, that data could also be used and could be justified. Okay, for your studies. Okay. So who are the individuals going to be responsible? The principal investigator, other than but the study director, the principal investigator, the veterinarian, the person in charge of the animal house, the technicians who are working along with the researcher, the committee. Everybody is responsible to actually review these endpoints, because we cannot just say I'm going to induce something and I'm going to collect so many times the frequency is this many times, so many withdrawals within this 1 hour. So all these things will have to be taken care. Okay, and what is the right technique to withdraw blood? For example, blood withdrawal is one of the very important endpoint. And when you are supposed to terminate. Okay, so as I mentioned to you earlier, what are the resources that you can rely upon to come to an understanding or determine this endpoint for your respective studies? Literature surveys, physiochemical tests, molecular modeling, in vitro tests will help you to give you some idea about the compound that you're working about, the drug that you're working. So sometimes there is some toxicological profile which are already established. So all these data will be very much important for you to move forward. Okay. So the next thing is how you identify. Okay, physical appearance, any alteration in the physical appearance, any alteration in the behavioral signs, any alteration in body weight. Okay, when you measure the clinical parameters when compared to the normal, how is this animal behaving? Okay, the complete condition, you will have to have a checklist and you will have to take note of all these things. So that will give you, okay, there is a drastic shift when compared to the normal. So it is not advisable to go beyond this one. Whether it is necessary for you to go to that endpoint, to that level, or to have the study extended to that point, it is very important for you to determine. So if you are able to collect your data and if you can close the study, please go for it. Okay, it is not necessary that you need to wait until that point. Okay, so then about making an informed decision to humanely kill animals or sacrifice animals. So the decision to actually humanely kill the animal must be made with appropriate clinical judgment, considering the severity of the condition, the amount of pain or distress, the prognosis, and the potential loss of valuable data. Okay, so the most clinical signs are not sufficient by themselves to support a decision to humanely kill the animal. So it is very necessary that you will have to depend on the possible the indicators, the useful indicators that I've listed here. Okay, so these are some of the very useful indicators when you will want to decide, okay, whether you want to sacrifice the animal or not. So under all these conditions, if you are going to experience this during your experimental work, then you might have to adopt the humane sacrifice protocol, okay, without any hesitation, because you need to minimize the suffering. Okay, so all these things are examples of the severe pain and distress, okay, experienced by the animal. So it's very necessary that you take into consideration all these sort of observations. So that is why if you are working with animal models, observation is very, very important. You cannot just say, because the animal cannot speak. The animal cannot verbally tell you what exactly is happening with it, right? So you need to follow these observations very, very carefully. Okay, so of course, when we are adopting to protocols that follows usage of anesthesia, you need to be very clear, or you need to be very accurate, because special considerations or characteristics will have to be taken into consideration, because the species characteristics and variations must be kept in mind while using an anesthetic. Because side effects such as excess salivation, convulsions, excitement, disorientation should be prevented and controlled while you are using this anesthesia. Okay, and this should be carried out under the expert supervision. Okay, where you are using anesthesia, you cannot just go ahead unless and until you are trained. Okay, so you need to take into consideration. The animals should remain under veterinary care till it is completely recovered from anesthesia or any sort of operative stress. Okay, so that is very important. So this table gives you the list, the list of the different compounds or the drugs that are used. Okay, and what is the dose that has to be adopted across different models. okay, when you are going ahead for the anesthetic procedure. Okay, so as per the literature, or even in my experience when I work on animal models, I rely mostly on ketamine and xylazine, but it is a scheduled drug, so you will not be able to just get it right away like that. So you might have to get a letter or something like that, a letter from the higher management of for example, the principal in the case of institutions, or any institutional ethical clearance supporting document to actually procure these sort of anesthetic agents, and it should be used with caution, okay, very carefully, okay, sensibly. All right, so methods to be adopted for human killing. For example, we are talking about euthanasia, right? It's chosen on the basis of animal species, the size of the animal, the excitability, presence of painful injury, etc. Suitable physical controls of animal being sacrificed is critical for satisfactory euthanasia to minimize fear, anxiety, and pain. Because while you're adopting that protocol, you should ensure that the animal that being sacrificed shouldn't be stressed, and you shouldn't be stressing the other animals in the group or in the environment as well. So the technique should be clear, okay, and it should be performed by an experienced staff. So training is very, very important. Okay, so the euthanasia should adopt all these things: lack of pain perception by animal, rapid, should be fast, okay, and it should be irreversible, very minimal interference with the experimental protocol, etc. Okay, and also the safety of the individual adopting. So these are the different agents or methods that are actually adopted for sacrificing animals, adopting the ethical practice. All right, so this one is species specific based on the species. The earlier one is species and what are the remarks, and now different species, what is the most appropriate agent or method. Okay, you have inhalation, and then you have physical methods, okay, intravenous methods, etc., etc. So here it has clearly stated there is some caution while we are executing the cervical dislocation. Okay, cervical dislocation, where we talk about the decapitations, right? So cervical dislocation, when you try to dislocate, then the weight of the animal should be taken into consideration, specifically when you are adopting the CPC SEA guidelines, right? And finally, to the last component about using experimental alternatives. So we have spoken to a greater extent about replacement. Okay, so that replacement comes in many forms. Okay, so we have well-established tissue culture methods. We have cell lines, the one that I mentioned earlier, primary cell lines, immortalized cell lines, explant cultures, we can call it as. Several other recent strategies, okay, have been made available to curtail animal experimentation, and not to forget, and also to simultaneously achieve improved effectiveness or efficacy of the data, okay, that are collected. So while alternatives to animal experimentation may reduce research dependence on animals, they currently cannot replace animal testing altogether. So that is why we need all these ethical practices to be adopted, at least to minimize wherever possible. Okay, so that is the ultimate goal behind having this at the or being having it cascaded to the institutional level. So these are we are not going to go in detail into these methods, but just for your information, some of the very recent advancements, which we call it as organ on chips. Okay, so where we have technological advancements where we would need the usage of microengineering, okay, systems, which will eventually be able to support living cells. Okay, so these organ on chips, or in short OOCs, are micro-engineered biomimetic systems containing microfluidic channels. So they have channels lined with human cells, which replicate key functional units of living organs to reconstitute integrated human organ level pathophysiology in vitro. So you are using technology, you are using engineering, and you are using human cells. Okay, so while you are directly extrapolating your data, okay, with human origin cells, then it is more valid. You might not go back to the preclinical sort of evaluation where you need animals to be used, okay, because you already have incorporated the human cell, so the data is much more supporting to further be translated. Okay. Another example where human skin equivalents are used. Okay, this is this model is to be adopted for autoimmune disorders or other cancer research. Okay, so based on the quality of the data, it can they feel or it is understood that this model is much more appropriate and sometimes could lead to replacement. Okay, possible replacement of animals. Okay, so in silico methods, of course, we do have some advantages now, or great advancements in the software or tools which could eventually go into the docking, tell you the microarrays, the RNA techniques that are the PCR techniques. Of course, it has been there for some time, but now with the help of very recent technologies or systems or the advancement in engineering, we will be able to come out with some valid data using the computer itself. So that data will be very much helpful for you to develop a protocol, a research protocol for the purpose of application for funding, because you have some experimental data which supports you very clearly, yes, this is the protein that you want to target, this is the molecule, whether the protein can bind to this molecule or not, or whether the molecule is larger, what is the size of the molecule, okay, what are the several points where you can actually target, which region of your chemical compound will have to be manipulated further to increase the safety of the drug or to reduce the toxicity. All these things can be done in vitro or using the in silico methods. So so that comes to the end of my presentation. So in conclusion, animal research cannot be completely neglected, but ethical practices will have to be adhered, will have to be followed, will have to be monitored and regulated wherever possible to ensure that the research can sustain. Of course, there's always a combat between different groups. Okay, for example, researchers and the welfare, but adopting the ethical principles framed by the different governing bodies in different countries will help, okay, to move the research forward. All right, so with this, I hope that my session was useful to all the budding researchers, and thanks the organizing committee for giving me this opportunity. Thanks.
Thank you, sir, for your valuable and informative session. And it was an excellent speech on ethical consideration in animal experimentation and research to reach the unreached, which we enjoyed thoroughly. A few questions from participants' side, seeking your guidance, sir.
Yes, please.
Thus, brief the role of experiment animals in virology study.
In virology study, so are you asking about the the role of the investigator? Hello. Can you explain a bit further what you are looking for in that question, please? Are you trying to ask for the roles of in which you are
Yes,
working on virology?
Yes, sir. Yes, sir.
Okay. From the pharmacology perspective, I would view it as taking into consideration the appropriate model. Okay, because working with animal models, using one example, now the current trend on vaccines. Okay, so there are several publications which is coming up whether to embark on animal models for studying this virus. It is not necessary that you embark on those animal models immediately, but you need to have adequate amount of preliminary data, as I mentioned to you earlier. So every every research will have to be approved. So you will have to anyway go through the ethical approval process. But there is something called as biosafety because using animals and when you're using microorganisms, every institution, every hello, sorry, yeah, okay.
Yeah.
Every institution will have this a protocol and it will have a component on the biosafety aspects. So you might have to comply with the biosafety aspects while you are working with microorganisms, okay, be it bacteria or be it virus. Okay, so you will have to provide adequate amount of information on the sources, how you are going to maintain and how you are going to do it, whether it is attenuated or whether it is still what form, etc. So in addition to your protocol, which is going to talk about the animal experimental part, you need to put an annexure where these biosafety principles are adopted. Okay, so that is the practice while we work on these microorganisms, especially.
Second question is why zebrafish is used in pharmacological research?
Okay, as mentioned earlier, why fish is uh, because the genome has been cracked already. The advantage is the animal model is smaller, reproducibility, availability is much more easier. All right, but although it is zebrafish, we cannot take it live that it is what to say. It is also live, so it also has to undergo. For example, you would have heard about fruit flies, okay, which is a tremendous advancement in research. The fruit fly, you would know the size of the fruit fly. So that genome has been cracked already. So which is giving what to say, very huge insights in technological advancements related to the genome level, okay, RNA, DNA level. So similarly, when this genome is cracked, you know which particular region is actually or how it is mimicking the human thing, the human model. So instead of going to preclinical using animal models and then translating into the human the clinical phase, you have more amount of data, valid data, which will be easier for you to translate. The translation aspect has to be taken into consideration. Otherwise, there is no point in doing research. Whether if you are not, to be very frank, not all the research can be translated, but they give you some valuable amount of data. So the appropriate model, these are all current techniques. These are all current upcoming techniques. I'm doubtful whether it has been widely practiced, but that is the reason, the current trend moving forward. It is because of the similarity, the greater extent of similarity of the human genome and the zebrafish. Yeah,
Thank
that's it.
you, sir.
Yeah.
Dr. Pradhan for addressing of thanks. Thank you.
Thank you, Myron sir.
All right. Thank you, sir. Thank you.
It's a great honor and privilege to propose vote of thanks on this memorable occasion. On behalf of management and staff, I sincerely thank Dr. Myron for gracing today's webinar. Thank you, sir, for your interesting and valuable information.
Thank you, Professor. Thanks for welcoming me.
And I would like to extend my sincere thanks to our management and our beloved principal madam for the constant encouragement support in conducting this webinar, and my heartfelt thanks to the participants uh, for making this webinar successful, and also thank the heads of various departments and faculties for their valuable suggestions, guidance, and encouragement in all our efforts. And I also to thank the participants for making today's session a success. I thank you one and all. I thank each one of you for making this event a grand success. Thank you.
Thank you. Thank you once again.
Thank you, Mayuran. Very informative.
Thank you, Professor. I will have to, I had to cut down a lot of information because of the time, because the information will have to be just right for the students, I think, for the BPharm and masters. We cannot go.
It was very nice.
Yeah, thanks.
Thank you very much.
Okay, sir.
Okay, bye.
Thank you.
Thank you.
Yes, sir.